Okay, so what this is essentially our approach. So we've been building these diagrams for a number of years, and of course people come along and say, well, it's only a picture. What else can you do with it? You know, where's the time? Where's the interaction? What more can you do with this? And to be honest, we had to shrug our shoulders and say, well, actually, it is a diagram. It's really useful, but everybody thinks it's really cool to model dynamics. Okay, so we thought, how can we do this? And I looked around at various approaches, and certainly these mathematical approaches that I described earlier were never going to work. The diagrams are too big. We don't know any of the constants by which things interact together. We just don't have the information, and the scale in which we're building these pathways is too big to even consider mathematics. And of course, as I say, the mathematics himself is a challenge in its own right to construct these things. So what we wanted to do was explore the possibility of taking these diagrams and to model their activity, as we might think about, you know, making a system that actually where we could think about the kinetics of how that little feedback loop might work, okay, for instance. So, as I mentioned, there are a lot of mathematical ways of doing this, and they basically break down to sort of equation-based, and the ordinary differential equation is one of the most commonly used approaches. There are many different types of ordinary differential equation, so there's different flavors of what you might use in this way. And basically, when you run a series of equations, you always get the same answer. So when you solve a series of equations, the answer is the same. Now, we kind of know that in biology, we do the same thing to a bunch of cells, and we don't always get the same answer. Or that we look at the cellular level, and each cell is slightly responding differently to a perturbation that we might make. And so this idea that everything is a fixed answer isn't quite what we want to do, and certainly it also requires parameters and is computationally intensive. So stochastic simulations are a way that we're actually modeling not only relationships, but actually the idea that these relationships might change, that there is a stochasticity. And so does anyone know what stochastic means? Randomness, okay? So randomness that we find is kind of randomness in biology, and to build that into the model. So we're really looking at individual reactions rather than the system as a whole, and it doesn't require parameters, and there's a number of different approaches. But it was these kind of starting assumptions. So in any of this kind of modeling world, you have to have assumptions. Okay, assumptions are very important. And so that we are saying that the rate of any given reaction in vivo is not known and cannot be determined. Now, they can be determined, but it's really hard work to do it. And even once you've determined that reaction, it's often in a test tube, not in a cell, and therefore how things work in a test tube is not the same as how they might work in a cell. So even though you might spend a lot of time getting the reaction rate, it probably doesn't matter. And even if this was not the case, i.e. that we can actually determine the reactions, then individually these factors have little effect on the dynamics. Okay? So we're assuming that even though we might be able to determine rate constants of individual reactions, that overall we don't think that these rate constants have a huge impact when taken as a system as a whole. So the reactivity flow through of pathways is primarily determined by the connections between the components, i.e. the network topology, and the amount of a given component. So we have just drawn the topology of a signaling network. And if we didn't have interferon there, then obviously you wouldn't get any activity. So, and the more interferon we might have, the more activity or more signaling that we might get. So we've drawn the connectivity, that's the important thing, and the one thing that we might consider as important is the more that we put into interferon, the more we might get in terms of a response. Seems quite reasonable to me. The pathway network forms the basis of the model, and the amount of any given component can be measured or inferred. So we can measure things quite easily, as we've seen with transcriptomics. We can measure the amount of gene very accurately in many cases, and that is sort of information that we might be able to feed in such a model. We can do proteomics, we can measure proteins. So we can, in this way, a lot easier and on a genome-wide scale, measure the amounts of things. It's a lot harder to measure the activity of things. So we also assume that biological systems are stochastic, i.e. one whose behavior is non-deterministic, so that we might do the same thing on 2 different days and get a slightly different answer, or a series of cells within a dish might all respond slightly differently to this thing. So it's the randomness of biology which is evident there. And of course, with the other thing we must bear in mind, which is the important thing, is that the model may be incorrect. There's an old saying that all models are wrong, but some are quite useful. So a model is a model, it isn't reality, it is a construction that we have made, and we should never forget that what we're actually representing is a model. Okay? And these assumptions may only be approximations of the truth. So we have to, that's our caveat, that's our get out clause for anything that we might do now. Okay? That this is just a model.